Sir, go ahead.
So shall I start the talk?
No. I'll give an introduction. I mean, I will introduce you to the audience, and then
Okay.
you can.
Right.
Good evening, all. My dear faculty colleagues and dear students, welcome to our 12th lecture in the invited lecture series. Today, we have another eminent person to deliver the lecture, Dr. Krishna Prasad Katuri. Currently, he is working with KAUST, King Abdullah University of Science and Technology, KSA, as research scientist in the Water Desalination and Reuse Center. He did his master's in Environmental Science and Technology, Jawaharlal Nehru Technological University, JNTU, and then he did his PhD in bioengineering from IICT, Indian Institute of Chemical Technology. Then he received Marie Curie Fellowship and worked with Newcastle University, UK for 2 years. And then he moved to National University of Ireland, and where he worked as Marie Curie experienced researcher in the Department of Microbiology and senior researcher in Department of Chemistry. Then he joined at KAUST as postdoctoral fellow, and from 2014 he is working as senior researcher in Water Desalination and Reuse Center. He received many awards, including prestigious Marie Curie Fellowship, and that even he received it twice, and Saudi Basic Industries Corporation Outstanding Research Fellowship, and Best Innovation Award for Technological Advancement from the International Society of Microbial Electrochemical Technologies. And his research focuses on underpinning anaerobic microbial oxidations for sustainable resource recovery from wastewaters based on electron transfer between electroactive biofilms and carbon surfaces. Dr. Katuri is a successful and active scholar with more than 60 international research publications in high-impact international journals and 8 international patents. And he is one of the world-renowned electrochemical, scholar in the field of microbiology and microbial electrochemical technologies. His research has been well received in the scientific community, and more than 33,800 citations and 32 index. Today, he will give a lecture on microbial refinery for conversion of waste to resources. I invite Dr. Krishna sir to deliver the lecture.
Hello. Thanks, Shabil, for your nice introduction, and good afternoon to all. Today I'm going to present my research topic on how a special group of microbes which are present in nature useful. We can use them to engineer biotechnologies for harnessing energy and water from wastewaters. So the relationship or interdependent relationship between two resources, that is water, energy, we call commonly we call that relation as water energy nexus. If you keenly observe these graphs, one thing we can realize that we are all living in a thirsty world for resources like water and energy. Predicted upward trend in population growth, energy and water consumption, warning that global consumption of renewables grow several orders of magnitudes in the future. As the consumption of renewables rose, global stress on water and energy will grow. If any imbalance happened between the water energy nexus, which can create tensions over food production in the future. So water, energy, food are the pillars upon which society can further advance, and the interdependency between water, energy, and food production and consumption is termed as water, energy, food nexus. The relationship between these components illustrated in this image, and you can see the strong symbiosis between these components in the society. Water is essential for energy production and conversion, and electric power generation accounts for essential, like accounts for around 15% of total global freshwater withdrawal for energy production. So similarly, energy is consumed to provide water for domestic, industrial, and agricultural applications. Same way, we consume both water and energy in order to grow crops and harvest crops, clean and prepare food. So on a global basis, food production is responsible for approximately 6% of total energy use and around 70% of global water use. So waste is also included in this diagram because the volume of waste can have a major bearing on nexus balance. So it is estimated that around 1.3 to 2 billion tons, that is equivalent to 33 to 50% of global food production, are lost or wasted annually, and finally it is ended up in dumping yards. Why? Because we consider it as a waste, but it is not a waste. It has, we can consider it as a source for generating resources. So because some of these wastes are recyclable because it contain a lot of organic rich biodegradable components. So the recent developments in biotechnology really useful for turning this waste to a resource. So if you observe in urban society, each house generates both liquid and solid waste, right? And these wastes are rich with biodegradable organic. So we can subject it for microbial remediation process, and most of the cities has infrastructure to collect and transport these wastes. So urban environment is the ideal place for implementing a waste to resource concept, because huge amount of solid and liquid waste generated every day in the urban society. So we can generate energy from solid waste in the form of methane gas, and methane gas can be used as a fuel source in automobile industry. This is well developed technology, and technology means harnessing methane from solid waste using anaerobic process. It's a well-developed technology you can implement easily anywhere and in any society globally. So, but I'm not talking about solid waste management in urban society. Rather, I focus more on liquid waste. So we technically call liquid waste generated in urban society as sewage or municipal solid waste, municipal wastewater, and which is low in organic content, however rich in nutrients such as nitrogen and phosphorus. So if you look at urban society point of view, how wastewater treated on regular basis. So we collect all sewage from each house and we dump in one place where they aerate this wastewater with oxygen. That's how they remove the pollutants from the wastewater. This is the common practice, and we call this process as aqueous sludge technology. So it is served for us more than 100 years, And the main purpose of this process is to remove the pollutants from such wastewater, such as carbon, nitrogen, phosphorus. And there is no doubt that after such process resulted in remarkable advances in wastewater treatment and standards of living and production of the environment from the pathogens. However, so let me explain what exactly happening after such process. As shown in this image, there is a tank. We fill the tank with wastewater, then we aerate them to provide oxygen. So there are certain groups of microbes which are abundant in nature. They grow together to remove the pollutants from the wastewater. So as I said before, we have carbon, different types of carbon, and nutrients such as nitrogen and phosphorus components. So we have to remove them before we get discharge this wastewater into the environment to protect the nature. So when you aerate this wastewater with oxygen, there are a group of bacteria, we call them heterotrophic bacteria. They oxidize organics, whatever the form it is, they oxidize them to CO2 and generate electrons, and they donate that electrons to an electron acceptor, which is abundant in nature, that is oxygen, to breathe naturally in the environment. So this is how they remove the organic pollutants from the wastewater. Similarly, there are other group of bacteria called ammonia oxidizing bacteria and denitrifying bacteria. They work together to remove soluble nitrogen components present in the wastewater to nitrogen gas. So if you look at the image here, one group of bacteria, that is ammonia oxidized bacteria, oxidize ammonia to nitrate. So here they use oxygen as a terminal electron acceptor to drive this process. They consume oxygen. And second group of bacteria, they convert nitrogen to nitrogen, nitrate to nitrogen gas in the absence of oxygen. So finally, one aerobic bacteria, one anaerobic bacteria, they're working together to remove nitrogen present in the wastewater to nitrogen gas. So similarly, there is a special group of bacteria which is called phosphate accumulating bacteria. What they do, they assimilate this phosphate to a polyhydroxy alkanite. It's called PHA, and we can use this PHA as a bioplastic for making materials. So these bacteria are responsible for removing the phosphate present in the wastewater. So they are working together to remove the pollutants from the wastewater, but how to design parameters to make them happy to work together is a challenging. So that's why we engineers and scientists work together to develop parameters to make them happy to remove the pollutants from the wastewater. So let's look at the water flow in a society. We are spending enormous amount of energy for producing potable water from natural water sources. Then we use this clean water for our daily needs like cooking, toilet flushing, bathing, and some other applications. And during this process, we generate dirty water, and we call this as sewage, right? Again, we recycle, we rely on energy intensive process for treating these wastewaters before it released back to the local water bodies. However, to resolve the future water energy crunch, one option is to recover valuable commodities like water, energy, and nutrients from the sewage. So smart integration of biotechnologies to the waste management systems will become resource recovery centers, which enable us to lower the cost for production of water. So that way we can offset the demands for high quality water. energy production that offsets fossil fuel demands, and nutrient recovery that offsets the fertilizer demands for the food growth of crops. So environmental scientists and engineers working on developing sustainable biotechnologies to engineering microbial communities to turn wastewater treatment plants as a microbial refineries to produce water energy from wastewaters. So as shown in this image, the current practice we use for wastewater treatment is aqueous sludge process. And if you look at this image, you can clearly observe it requires huge area that needs more carbon footprints, and the energy consumption is high. We typically use around 1.6 kilowatt hour of energy for treating meter cube of wastewater treatment. However, we are entering in a new era in wastewater management cycle where we think of used water as a valuable resource for water energy nexus. So, however, there is a growing awareness that the current paradigms of wastewater treatment are no longer adequate to meet the demands of rapidly growing human population and urbanization. Thus, environmental engineers slightly modified the existed aqueous sludge process by integrating the aqueous sludge process with porous membranes, and we call this as aerobic membrane bioreactor technology. This is the current state of technology for wastewater treatment and reuse. And as shown in this image, there is a porous membrane we place inside, and it can filter the clean water for reuse. However, two dimensions of this process are energy intensive and fouling of the membrane, which requires frequent cleaning or replacement of the membrane, which increases operational and capital costs. So these technical issues associated with aerobic membrane bioreactor is the most important driver on the development of future wastewater treatment technologies. So here you can see a cattle tummy. The reason I want to project this image is cattle tummy is a perfect example for anaerobic digester. Formation of hydrogen and acetate during grass fermentation in the cattle's digestive tract provides a perfect niche for the localization and growth of methanogenic bacteria to generate methane. And these are special group of bacteria. They grow in anaerobic environment where oxygen is absent. And they turn hydrogen and acetate to fuel, that is methane. So the bioprocess insights from the ruminants' digestive tract inspired the scientists to develop a anaerobic digestion biotechnology for harnessing methane gas from waste. So in the anaerobic digestion, what exactly happened? A group of bacteria work together to sequentially transform waste into a methane gas. As shown here, this is well-developed biotechnology, however, which is suitable for high organic strength wastewater, such as wastewater produced from molasses industry, or food waste industries. However, we can't use this technology for recovering energy from dilute wastewater such as sewage produced in the urban environment. So now it is the time to think outside the box to develop solutions for recovery of both water and energy from low organic strength wastewaters. So at our institute, we are conducting a goal-oriented fundamental research to optimize and create novel biotechnologies for wastewater treatment and reclamation from the sewage, and we should be robust, scalable, and capable of providing tailored water quality with minimization of energy, resource, and carbon footprint. We are we are guided by the vision that convergence of environmental biotechnology, microbial ecology, and material science will provide viable solutions to the three greatest challenges facing society in 21st century, that is water, food, and energy. So this integrated multidisciplinary research field called as electromicrobiology and related biotechnologies named as microbial electrochemical systems. As shown here, we use various omic tools such as genomics, proteomics, transcriptomics to study how these bacteria working together to turn waste to a resource, because we need to understand how to enrich them, how to make them work together, how to maximize their efficiency is matter. So we study the function at the genetic level. We extract mRNA, we extract DNA, we extract proteins from these cells to study how they are working together. Okay. So as shown here, there are certain group of bacteria they existed in the nature. What they do, they oxidize organics presenting in the surrounding nature to a CO2. During this process, they liberate electrons, and these electrons reduce iron to iron 3 plus to 2 plus. So during this process, they gain energy. That's how they breathe in the nature. So iron oxide is very rich in nature, and these bacteria, they can use them as electron acceptor to survive. And we call this bacteria as metal reducing community. So here you can see how this bacteria mineralizing or solubilizing insoluble iron oxide in the nature. See these cells swim towards these insoluble particles, and they use them as an electron acceptor, and this how they solubilize, because once it's reduced iron oxide, it turns to +2 form of iron and then solubilize in water. That's how they breathe in the nature. So we consider the special bacterial extracellular electron transfer physiology as an opportunity to engineer microbial electrochemical technologies for harnessing energy from waste. In this process, these bacteria use what we do. We place these cells into airtight glass cell, and we place carbon electrode as electron acceptor, and we apply potential, and these bacteria swim towards this anode and grow as a biofilm to tunnel whatever the electrons produced during the substrate oxidation towards anode. This is how we make them happy to grow on the electrode, and we use these electrons to reduce as energy at cathode. So here you can see how this bacteria swimming towards anodes to form as a biofilm, and then they transfer electrons externally to the anode, as shown in this illustration. The next is, you can ask me how this bacteria can transfer electrons to the insoluble electron acceptor. So they can form as a film around this anode. During this process, they express membrane-bound redox electrically conductive proteins, which can wire cell to the anode. So that can make electrical bridge between cell to the anode to transfer electrons from cell to anode. And another approach is expression of electrically conductive nanowires. So these bacteria, some of the bacteria from this community, they can express metallic conductive like nanowires. They can express in centimeters long to turn electrons from cell to anode. So in this process, they're not necessarily bound to the anode surface. They can turn electrons from away from the anode surface too. So this is the beauty of this community. So then the next question is why we have to use this community. These electrochemically active bacteria are ubiquitous in nature. So you can take soil or fresh water or seawater sediment and inoculate in the reactor, you can enrich them easily. So that's why we are interested on these technologies using these special microbes. And they grow very fast, and substrate affinity is very high, they're very robust, and they grow with time, so you don't need to waste a lot of money every time to inoculate the cells, and it's controllable. We can control their physiology externally using some external parameters like applied voltage or temperature. This kind of parameters we can use to control their physiology. And we can start them very quickly, followed by adding inoculum into the reactor. They grow very fast. Within 2 to 3 weeks, they grow as a biofilm. They can generate current. They stabilize easily. So using this extracellular transfer property of this bacterium, we scientists developed two novel biotechnologies. We call them as microbial fuel cell and microbial electrolysis cell. As shown here, in microbial fuel cell, these bacteria grow as a film on the anode, and they generate electrons. That electrons transfer to cathode, where they reduce to water in the presence of oxygen, and finally the circuit closed, then we'll get the electricity. So in this process, we can generate bioelectricity from the organic waste. Similarly, we have another process, we call it as microbial electrolysis cell, where the anodic reaction is same. However, we reduce electrons, protons, which are generated at the anode, to hydrogen gas at the cathode. So this way we can harness green energy, that is hydrogen, from organic waste. So this is the beauty of this technology, because hydrogen is a green gas. Now throughout the world trying to use this as a fuel for automobile technology. So then somebody can question me, well, there are technologies which are available throughout the world. Why can't we produce hydrogen from the water instead of microbial electrolysis cell? In traditional technology that's called hydrogen hydrolysis, what we generally use, we use pure water for water splitting, and we use expensive anode catalysts. They're generally platinum or iridium based. They're very expensive, and we required high energy input, that is around 9 to 12 volts, to split water to hydrogen gas using hydrogen electrolysis. However, in the MEC technology, we use wastewater as an electrolyte, And the anode catalyst here is bacteria. They're free of cost and they grow with time. So we don't worry about the catalyst life. And second, the energy input to drive this process is very low. That is around 0.7 volts. This is almost 10 times lower than traditional water hydrolysis technology energy requirement. So that's why my research, I'm so passionate about this technology. However, with MFC or MEC technology, we can harness energy, but we can provide water needed for reusable application. So my research focus on MEC, but I would like to maximize resource recovery from these technologies too, not only energy. I want to recover water too. So what I have to do? So what we did, if you look at the process point of view, we feed MEC with wastewater. Then these wastewater organics are fermented to propionate, butyrate, acetate, and hydrogen gas by a fermentative bacterium. Then this acetate consumed by this electroactive bacteria at anode to generate electrons, protons, and then they reduce to hydrogen gas at the cathode. This is how MEC works, right? So what we did is we replaced traditional benchmark cathode with a porous hollow fiber cathode architecture. We made this material with nickel, and the beauty of this material is it is porous, so it can filter treated water as a clean water. At the same time, it can act as a cathode for reducing protons to hydrogen gas. And we call this reaction as hydrogen evolution reaction. And we call this technology as anaerobic electrochemical membrane bioreactor technology. And we published a couple of papers on this topic. Please have a look for more details on this technology. As shown here, you see the treated water recycled as a reusable water, which is very clean, and the organic content and turbidity is very low, and we can use for some non-potable applications like irrigation and agriculture or cleaning aspects. So here you can see the schematic representation of anaerobic MER, and on the right side some images of nickel hollow fiber membranes, which is used as a dual functional cathode. Also nickel hollow fiber cathode architecture solve some other bottlenecks associated with this process scale up, such as how to improve the cathode to surface area, cathode surface area to volume ratio, and the scale up, the packing density issues. These are the technical problems we face with MEC. However, with our novel nickel hollow fiber cathode architecture, we managed to overcome these limitations, so we can scale up this technology at any time. Also, these materials are made up of nickel, which is very earth abundant, so it's easy to fabricate, and the cost of production is very low. So I'm very excited to show this video, the slide, because this has been the most thrilling research excellence experience in my life. My scientific discovery on anaerobic electrochemical membrane bioreactor concept selected to deliver a 20 minutes oral presentation to a renewed world entrepreneur and a philanthropist, Mr. Bill Gates. So followed by our lab scale experiments, we plan to plan to test some proof of concepts at pilot scale using real wastewater. So here you can see photographs of our pilot reactors. We are going to operate them soon using solar power as renewable source. As shown in this animation, the bacteria swim towards anodes to grow as a biofilm. Then they generate hydrogen on the cathode surface. So this is our technology animation. So as I mentioned earlier, in many parts of the world, development is very scattered, and many residents are not connected with central sewer collection and treatment systems, right? So our aim is to develop a decentralized systems for wastewater treatment and resource recovery, which are movable, plug and play, modular design, and robust in function. So as shown here, we can move this containerized technology to anywhere, and we can install quickly, and then we can recover fresh water. And this technology is scalable. And whatever the water we recover out of this process, we can use for gardening or irrigation purposes. So let me explain how our technology is useful in Indian context. In India, the urbanization and population growth expected to increase in coming decades. There is no doubt India is thriving now and will be in the future. So there is a huge demand for water, energy, and food expected, right? So in current scenario, 70% of our domestic wastewater produced in urban area not treated, and Indian government planning to build new smart cities and new infrastructure for waste management. So modernizing the waste management infrastructure with current trends in waste management, always useful for addressing the energy and water demand and improving the public health and food security in the nation. Finally, I would like to thank VIT AP University for giving this opportunity to present my work, and Dr. Shabil for arranging this virtual meeting and coordinating with me as our emails, and Dr. Banerjee Chekka for requesting me to give a talk at VIT and giving a chance to present my work. And finally, I would like to thank audience for listening my talk, and I'm very happy to address your questions. Yeah.
Thank you so much. It's a nice talk. And there are a few questions.
Mm-hmm.
One is, the student want to ask that if we have the first condition, water extracted from water pump, that's groundwater, and we boil it properly. That's one set of water. And in second, the water extracted from the same source, but this time it is purified from a RO or a purifier. So which is better with respect to the drinking water?
Well, RO-based technology is very expensive. You have to use expensive membranes, and you have to spend 2 kilowatt of energy for treating meter cube of groundwater to generate fresh water, right? But if you boil, and as long as there are no fluoride content, you can boil and you can use water straight away. That is the best approach. However, if your groundwater contaminated with metals or fluoride, my best advice is you have to filter them to remove the pollutants before you consume. So, example, Vijayawada surroundings, I believe groundwater is fresh. free of pollutants. So take groundwater, boil it, and use it. That's my advice. You don't need to go for RO.
Yeah, that's great. And you said the majority of wastewater is not treating. Then and your technology, how much water we can purify using that technology?
Well, it depends. That's why I told first we are concentrating on decentralized technology because if you look at India point of view, most of the places the sewer network not connected well, right? Example, take Vijayawada. Hello,
Yeah.
hello, can you hear me? Yeah.
Yes.
Yeah. So if you take Vijayawada point of view, Vijayawada not well connected with sewer system. Only the central part of the Vijayawada connected with central sewer system. So you can collect wastewater from the household. But if you go outside the Vijayawada, you don't have a sewer system. So the decentralized system for wastewater treatment is difficult. So we can place a tank, we can place a containerized treatment plant, and we can plug into houses and we can treat, right? But if you have more wastewater, what we can do a centralized system, or we can scale up system. Because they are plug and play systems. We can stack them together to treat more wastewater. Yeah. Volume. Depending on the inflow of the wastewater, accordingly we have to act.
Yeah. Yes. And any questions from any other questions from audience, students? Uh, I think there is no more questions. Yeah. At this moment, there is no questions from student side. We can conclude the session like this.
Okay.
Sabil sir,
Yeah. Sure. Yes.
I would like to Krishna sir. I would like-
Yes, sir.
Krishna sir, this is our HOD, Pramasyam sir.
Okay. Okay.
Welcome sir.
Listen, nice to meet you.
Uh, Thank you for your nice talk on the excellent subject. It's really interesting for us.
Okay.
I think you are merging the advanced technologies, microbiology, electrochemistry, material science, and
Yeah.
practical applications. That's a beauty in your this one, sir. Research sir can
Yeah.
talk. It's really interesting, fascinating for us.
Yeah. The reason is when Sabil contacted me, he said around 500 students participate in this presentation, and they're from different backgrounds. So then I thought I want to make my presentation a bit interesting, rather boring the audience. And these are the problems everybody facing right now in the society. That's why I presented my work in a way that it will explain the importance of science in society.
Sir, I'm having one small this one's clarification, sir.
Please.
Hello, sir.
Yeah, please tell me, sir. I can hear
Sir.
you.
Yes, sir. Regarding the biosafety, we are handling the bacteria and other things, sir, in home or in small micro scale level or medium scale level.
Mm-hmm.
If it is a class traditional materials, we can store and we not worry about the contamination or the degradation, sir. But here for biosystems, this is a biosystem,
Mm-hmm.
So how to
Yeah.
store and how to handle with biosafety everything? Is it any formulation you're having, sir, like that? You are going to have some advice, sir?
Yeah, actually, we use chlorination followed by extracting the water. We pump chlorine so we can kill the pathogens, including viruses. Before we extract the And second thing, whatever the biomass inside the reactor, we frequently as a life cycle, and we collect them, dry them, either use that as fertilizer. We can. Hello.
Hello. We can wind up the session. Shivaya, sir.
Yes, I can hear you.
Yeah. Okay. So
So shall I give vote of thanks?
yeah. Please.
Okay, sir. So very good evening, all of you students and my dear colleagues, and today guest speaker, Dr. Krishna Katuri. So thank you very much, sir, giving wonderful lecture today and spending time with our students and with us and giving a wonderful talk. As I said, our HOD sir, Pawan Sivam sir, this is a really interdisciplinary kind of research topic and greatly explained the applicability of microbiology and electrochemistry and biotechnologies, how really we can use this all the technologies, advanced technology in purifying the water, especially. So you give us students, please try to understand at this stage, but you can use this technology in future, so we can get the smart water technologies. So thank you, sir. You giving a wonderful lecture and today, and thank you very much, all the participants, and especially thanks to Professor Pravam Sium sir, HOD Department of Chemistry, and Professor Shabil, Coordinator of Environmental Studies, giving this opportunity. So thank you one and all.
Thanks for listening to my talk, and see you next time.
Yes, sir.
Yeah. Thank you so much, sir.
Yeah, thanks. Okay.
Okay, then. See you.